Welcome to my presentation. I am Snigdha Bharadwaj, and today the topic of presentation is microencapsulation. In the end of my presentation, you will be able to understand the basic fundamentals of microencapsulation technique in novel drug delivery system. So, let us get started. So, as you can see the slides, microencapsulation is a technique in which a relatively thin coating is applied to a tiny solid particles or liquid droplets to give a small capsule of multiple useful attributes. This means a small polymer coating is applied onto a core particle, let it be solid, let it be in liquid phase. In some papers you also can see that gaseous phase also entrapped in the solid polymer coating. So, here the technique involves the coating of the core, that means the active drug particle in within the polymer capsule. So, this technique generally known as microencapsulation because the encapsulation is done at micrometric level. Generally, it is used to encapsulate enzyme, cells and other proteins. Apart from pharmaceuticals, it also utilizes to encapsulate the biological materials like cell, enzymes, protein, peptides, macromolecules, we also say this, food ingredients, etc. at the micrometric levels. The product obtained by this process is termed as microparticles, microbeads, microspheres and microcapsules, etc. These are the terms we use for microencapsulated particles. The particle size of microcapsules ranges between 1 to 5000 micrometers. That means, the size of the particle lies within the micrometer range, not more than that. It is a well designed controlled drug delivery system which can overcome some of the problems of conventional dosage form. Why we opt these novel drug delivery systems? Because it has several advantages over the conventional dosage form. We will discuss later in detail. It enhances the therapeutic effects of a drug candidate, and to obtain maximum therapeutic efficacy of the drug, it is to be delivered through any of the route of administration to target the tissue into the optimal amount in the right period of time. So, these all three parameters give the leads to results in little toxicity and minimum side effects. So, through this microencapsulation, we can able to achieve the targeting effect of the drug molecule within the body, the optimal amount, that means the dose can be minimized using these techniques, and the time time can be increased or modified. We can get modified release profiles of the drug candidate. So, here as you can see here, this is the pictorial representation of microsphere, or we can say microcapsule. The picture represent the above layer represent the polymer coating, whereas the inner core shell represents the drug candidate or the active particle you want to encapsulate. So, this is written here, core shell containing drug. That means, the drug will be encapsulated within the polymer shell, or this layer represents the polymer coating. So, based on the size and manufacturing processes, we have variety of microspheres in shape, right. So, the ideal shape we always consider this mononuclear spherical for microspheres, but we can have during manufacturing, and depending on the variety of factor affecting the process of manufacturing, we can have variety of shapes of microsphere during the process. So, first one is the mononuclear spherical shape, which consists of a core and the coating material, right, single core particle. Whereas, in multinuclear spherical, you can see that variety of core particles encapsulated in a single layer of polymer, right, or we can say coating material. In multinuclear irregular shape of particle represent the outer layer of polymer is irregular in shape, not in spherical, and the multinuclear irregular cluster represents the cluster formation of polymer consisting the drug particle within it in the cluster formation. It is not uniform, but you can have these type of cluster during preparation of microspheres. Now, the dual walled microsphere represent the multi-layer coating of the polymer substance onto the core material. Understand? That is why we said it dual, because you can have dual layer of polymers. Now, encapsulated capsules means the single polymer coating contains already existing tiny microcapsules within the layer. So, here you can see that 3 microcapsules or 3 microsphere are entrapped within the polymer layer. So, these are the types of microcapsules you can have during the manufacturing process. Now, we will discuss about the fundamental considerations of these microencapsules. So, basically we use drug and the polymer apart from the other excipients during the preparation. So, polymer, or we can say the coating material, is the prime concern here. So, nature of core and coating material. If we say coating material, that means we are talking about the polymer agent we are using to coat the core particle. So, core represents the drug candidate or the active constituent we are using, and for which we are modifying the release using the different kind of polymers. So, nature of core and coating material is of important concern. Stability and release characteristics of coated material. Basically, the stability issue during the release, we are modifying the release, we are modifying the dissolution profile of a drug candidate. So, here the polymer type and the stability of the polymer agent is of great importance. So, this will directly affect the dissolution rate, dissolution profile, activity, therapeutic efficacy of the drug. And the third one is method of preparation. So, method of preparation is also one important factor to identify the yield, several factors like yield, the stability of the prepared microcapsule, the size of the microcapsules prepared by which type of techniques, or what type of size of microparticle you need during the manufacturing. So, it depends on the method of preparation as well. So, we will discuss it later. Now, first we will talk about the composition of the microsphere. As I told you, various other excipients can be used during the preparation, like organic solvents, stabilizers, diluents, but basic prime concern here is the core material and the polymer material we are using to prepare microencapsules. So, first we will discuss about the core material here. The core material is a specific material to be coated. It may be solid, liquid in nature, and in some papers and reported literature, you may also seen that the gaseous form is also entrapped in the form of microencapsulation. It can be a single substance or component mixtures like API, active pharmaceutical ingredient. Okay? Active pharmaceutical ingredients, and in the application you can also seen that it can use to encapsulate fragrances, it can use to encapsulate coloring agents, and variety of applications are there to encapsulate different. It is just not API in terms of pharmaceuticals, right. So, diluent stabilizers you can encapsulate it using this technique. The coating material are the polymer agents or the film forming polymers and must be cohesive with core material. That means, if the polymer would not be of cohesive nature with the core material, that means the encapsulation will not be appear in that case. If it is not cohesive, if the interaction between the core and the polymer is less, the microencapsulation will be affected. Non-reactive and chemically compatible with core material, these are the ideal properties of the polymer which we are using in microencapsulation process. That means, the polymer which we are using should be inert in nature, non-reactive and non-toxic. This means that the active agent we are using is a co-drug or the drug active component here, which has medicinal value. If polymer has all these reactive properties or toxic properties, that may hamper the release of the core material, or that may hamper or affect the therapeutic efficacy of the drug within the body. So, it should be inert in nature, but it should be cohesive side by side. Coating must provide strength, flexibility, impermeability and stability. All these parameters to be followed while choosing your polymer for the microencapsulation process. So, definitely if we are coating the API or the active constituent, so we may ensure that the particle particle forming in the process should be stable at particular temperature, at particular pH, at particular route, through a particular route which we are delivering. So, all these properties should be there to select the proper or appropriate polymer for the core material. Now, next slide represent the pictorial representation here of the core material, coating material and the vehicle which we are using. Apart from the core and coating, we use vehicle in which the solutions are generally formed while mixing the core and coating materials, right. So, here we can see in core material, as I have told you, that solid and liquid nature can be used of the drug candidate. So, mixture of drugs, in solid form we can use mixture of drug, the stabilizers, diluents, excipients, etc. So, we can use variety of solids while encapsulating the solid form, and in liquid form it either we can use the suspension form of the drug candidate, or we can use the solution form. Suspension or emulsion form is like two immiscible phases, separate immiscible phases, but one phase is dispersed into the other phase, and in solution the drug is solubilized in the system and the solution we can use directly to encapsulate within the polymer shell. Okay. Now, we will move to the coating material part. Here you can see various type of polymer categories have been mentioned, like water soluble resin. Examples are gelatin, gum, arabica. These are natural polymers we can use for microencapsulation, and PVP, that means polyvinyl pyrrolidone, CMC carboxymethyl cellulose, MC methyl cellulose. So, these are the various polymers which we can use for microencapsulation, and these lies under the category of water soluble resin. For water soluble kind of preparation we can select these type of polymers. Now, the water insoluble resins which we use for microencapsulation techniques are ethyl cellulose, polyamide nylons, and waxes and lipids. The third one category is paraffin, beeswax, stearic acid, stearyl alcohol, glyceryl stearates. All the stearates type we can use for microencapsulation comes under waxes and lipid nature polymers. The last one category is enteric resins, shellac. Shellac is a natural polymer which we use specifically for enteric reason, because the polymer degrades or erodes at the enteric pH only. It will not be changed or it will not release the drug in the gastric pH. So, here these are the various polymers which we can use for microencapsulation. Apart from that, there are variety of polymers which we can use for microencapsulation. Now, the vehicle may be of two types, aqueous and organic. So, depending on the type of nature of the drug and the polymer, we can choose the aqueous phase as well as your organic phase. Next, these are the prerequisites for ideal microparticulate carriers. So, some some considerations we give during the manufacturing process of microspheres, like longer duration of action. Why we are choosing these carriers, micro carriers for drug delivery system? Because we have some considerations there. Why? We want longer duration of action of a drug within the body. We want control on control drug release content in the body when once administered, increase in the therapeutic efficiency by decreasing the toxicity and the side effects. We can say that drug efficacy is increasing. Okay. Protection of drug, because the drug is encapsulated within the polymer shell. So, somehow it is protecting from the outside environment within the body. Biocompatibility, yes, polymer may be of two types, biodegradable and non-biodegradable. If we are using biodegradable polymers, then they will eliminate or excrete through the body without hampering the release of the drug. So, relatively stable, these preparations are stable as compared to your conventional dosage form. So, if we talk about novel drug delivery system, then we can have solid benefits and solid some considerations to which we can aim to develop the alternate therapy. So, these are the several considerations which needs to be studied during the manufacturing process. If the preparation is giving you the control release or not, if the stability is increasing when you compare this with the solid conventional dosage forms, if it is giving the stability and other parameters very well or representing the margin remarkable difference while preparing these alternatives, then we can always choose these type of preparation for the betterment of therapy. Reduction in toxicity, yes, direct interaction of drug within the organs, or it is not connecting directly with the organs. So, we can expect that the toxicity level, or we are reducing the dose as well. So, we can expect the toxicity will get reduced in that case if we give the drug in the form of this system, microparticulate system, and the water solubility. So, these are the various factors which needs to be considered while making these type of carriers. Now, move on to next slide. Factor influencing encapsulation efficiency. So, encapsulation efficiency is the most important parameter while we manufacture microencapsules, because how much quantity is encapsulating within the polymer shell is of prime concern here. If the drug, majority of drug is not encapsulated, is free, then what is the making of these microencapsulation? This is of important concern. So, we have represented two charts here, flow charts. These are the parameters like high solubility of polymer in organic solvent, less miscibility of organic solvent with water, low polymer concentration, high dP/Cp ratio, slow solvent removal rate will leads to slow solidification of microencapsulated particle, which finally resulted into low encapsulation efficiency. Whereas, the vice versa is low solubility of polymer in organic solvent, high miscibility of organic solvent with water, high polymer concentration, low DP-CP ratio, fast solvent removal rate will leads to the fast solidification of microencapsulation, microencapsulated particles, and finally, it leads to high encapsulated efficiency. So, our basic or first aim to get the encapsulated layer onto the core material. So, these are the factors. If we modify in that way, then we can have high encapsulation efficiency. Now, we will discuss about the advantages. Why we choose micro encapsulation technique over the conventional dosage forms. Conventional dosage form means the oral solid dosage forms like tablet, capsules. Okay. So, here are some advantages, like it is a new working tool for industrial pharmacists. Of course, these comes under novel drug delivery systems, and all the researches are going on to develop these dosage form as an alternate or as the main therapy for the treatments. New dosage form, microencapsules convert liquid to solid, example eprazinone. So, these techniques definitely encapsulate solid as well as liquid. So, that is why it is written here that they converts the liquid to solid form by encapsulating the polymer layer. Alteration in colloidal and surface properties. Yes, the alteration, the surface properties of the drug, because it is encapsulated in the polymer shell, and the polymer layer is the outer layer of the microcapsules, and the surface properties has been modified while administering into the body. So, surface area is increased and the release is modified in that case. Ability to control the release rate of drug. Will get the prolonged release rate of active components through these type of dosage form. If we make microencapsules, if we make microspheres, then we can expect the sustain or control type of release of the active components from the dosage form. Apart from this, every system has pros as well as cons. So, we will next move on to the disadvantages of the system. So, basically we have considered all the fundamental parameters or factors during the manufacturing, but after that also we can face some formulation difficulties. No single microencapsulation process is adaptable for all core materials, that means for all drugs. Core material represent here is the type of drug which we are using, not a single process has been established for all types of core materials. That is why we have multiple formulation methods to prepare microencapsulation microcapsules, because we have different kinds of drug materials which are having different kind of solubility, stability issues, temperature issues, degradation issues. So, we will choose the technique accordingly. Incomplete or discontinuous coating may occur. We said that the uniform layer of polymer is coating onto the core material, but sometimes inadequate or improper coating may occur during the manufacturing process. Of course, this may be due to the error in handling, error in process, some levels. So, if we modify or if we develop alternate approaches to that, we can overcome this difficulty. Sometimes failure to produce the predicted release rate. We say that we can have prolonged release rate or the modified release rate of the drug candidate, but sometime in some cases we cannot expect the adequate release, or we can have inadequate release. So, for that rectification we need to consider the various parameters which we used in the manufacturing process. So, that to overcome these factors affecting the process, we can have the proper or modified release. Economical limitations. Definitely the process of making microcapsules is not cheap, it is expensive, because we are using variety of ingredients like organic solvents and high level equipments. So, we can have the microencapsulation technique manufacturing is bit expensive. Now, why we are choosing these microcapsules over the conventional? What is the need of making all these modified or small level particles in the pharmaceuticals? For the pharmaceuticals, like improve powder characteristics of vitamins, for example, riboflavin, niacin, thiamine. So, these are the reported literatures which say that the powder characteristics after converting the raw form of the drug into the microencapsulated particles, we can have improved powder characteristics of vitamins. Reduce hygroscopic properties like sodium chloride. Sodium chloride is hygroscopic in nature, and if the moisture is present in the environment, it may liquefy. So, to prevent these hygroscopic nature, we can encapsulate sodium chloride into the polymer coating, within the polymer coating. Then reduce hazards, insecticides, so directly contacting. So, we can have this kind of approach to reduce the hazard or hazardous activities, prevent incompatibilities between drug, example aspirin and chlorpheniramine maleate. So, using this technique, we can prevent the direct interaction of the two incompatible drugs. So, this is a very good technique for this type of difficulties which we are facing. Reduce sensitization, example ampicillin trihydrate. So, because we are preventing the direct contact of drug to the system's organs. So, we can expect that the sensitization or the sensitive drugs cannot harm the organ directly. Reduce gastric irritation like aspirin, potassium, enhances the stability of fat soluble vitamins, protection of core material against environmental conditions, right. We are protecting the core material within the polymer shell. So, these all are the benefits through which we can protect our core material from the environment. Used to mask unpleasant taste of drug, definitely if the drug is unpleasant while taking orally. So, patient may show some sort of incompatibility in that case. So, we can mask the taste of the drug particle by this process. Production of sustained release dosage form. So, this is the important need here to modify, to get the modify or advanced release of drug. So, we can have the reduced frequency of dosage form in a day. So, this is the basic approach to make all these microencapsulated particles. So, applications are, it is used in vaccine delivery. Variety of applications are there that in which microcapsules or microspheres have been used. Microspheres interaction between macrophages at the cellular level, targeting using microparticulate carriers like ocular delivery, intranasal, oral delivery has been achieved. Magnetic microspheres have been used in chemotherapy to achieve the lesser toxicity within the organ. Monoclonal antibodies in case of immunotargeting, chemoembolism to block the particular target at the area, so that the drug can block the blood vessel blood flow, and the system microspheres can release the drug to the affected area. Surface modified microspheres, we can have gold, silver, copper coating onto the microsphere surface. Imaging microsponges, these are the advanced form. So, highlights are microencapsulation is a new technique to incorporate coat the active ingredient, right. So, it is a new technique, and this technique is being used by various industries to encapsulate or to make fragrances, food industries, pharmaceuticals and other industries. So, the success outcome of this technology depends on the correct choice of coating material, the method of preparation and the pattern of coating material, of the method of preparation and the stability issues. So, I hope you get the basic understanding of the topic. Thank you. Stay safe.